Okay, guys, we're going to get started. So I'd first like to welcome all of you to Iowa State, and thank you for coming. We've got about 100 folks registered. There'll be maybe 25 or 30 more from Iowa State who show up tomorrow morning. Understandably, they're on their couch watching Netflix tonight, since they've heard me before. So this is a NEFA-funded workshop with a focus, as you all know, on genomes to fields. So the title is here, High Throughput Field-Based Phenotyping Technologies for Genomes to Fields Initiative. There are really two major goals. I mean, one is to just promote discussion among ourselves, but really to look at different field-ready or near field-ready high-throughput phenotyping technologies, talk about different possibilities, and also begin to think about what we want to do with the G/E project in terms of getting technology out into those sites. And then NEFA is very interested, AFRI is very interested in having a white paper where we outline really what the needs of the community are. Carolyn Lawrence has very kindly agreed to organize and take the lead on two sessions where we begin to draft the white paper. One will be tomorrow late afternoon, and the other will be Tuesday morning. And there is a URL on the Genomes to Fields meetings, facts, workshop. So if you tunnel down from the Genomes to Fields website, there's a URL there that will take you to sort of an outline of the white paper and some discussion topics and maybe even assignments of who is going to be in which groups tomorrow. So I encourage you to look at that tonight when you get back to your room and you can't sleep. All right. So we have a very, very full schedule. We'll have a full day of presentations tomorrow on data collection, analysis, interpretation, and new phenotyping technologies. There's going to be a lunch panel focused on entrepreneurship, which I think you'll find quite fun. And then a late afternoon panel focused on what it's like to work in industry for grad students. And we'll also have this first session working on the white paper. And then we'll have an evening poster session after dinner. And then Tuesday, we've got a half day scheduled of presentations and more work on the white paper. And then an afternoon visit for those who are signed up in advance for the tour of Pioneer's high throughput phenotyping facility down in Johnston. So with that, I'll introduce our first speaker. And what I'm going to do is really talk about some ideas with a few examples of field-based phenotyping technologies. I'm not going to talk too much about analyses. I took those out, but I will highlight some speakers from tomorrow who will mention that. And then I'll also finish with a section on community building, which is something that I've thought about a lot and I think is something we need to spend a lot more energy on. So I'll begin with this slide, which some of you have seen before, but it really sets the stage here, the challenge. So on the left, the green panel there is corn grain yields. You probably can't read the x-axis, but you've all seen these before. That's time. The y-axis is grain yield. The inflection point is in the mid-1930s. And who knows what happened in the mid-1930s? What drove that inflection point? There are a couple of correct answers, and there are a couple of wrong answers. Hybrids, that's one. What else? Fertilizer. What was that? Single cross, that's the hybrids. Inputs. Others? What? Machinery, that was more later. It was statistical plot design, actually. People were trying to breed without taking into account the variability of soil type. But that curve has been moving up fairly steadily. And the reason it's been moving up fairly steadily, and it's hidden, I've drawn that red line on there. The original graph has a series of different regressions as new technologies are introduced. But what's been driving that up is, in fact, the introduction of new technologies every decade or two. The middle slide is for audiences not like this one, but when I talk at schools where they don't have a real ag presence or they're outside of the Midwest, that's what happens when we have a good year and there's not enough room in the elevators for all the corn, right? And on the far left there is my wife and younger son to give you a sense of scale. So working together, the breeders and the agronomists, the seed companies have just done an amazing job. The challenge is keeping that curve moving in the right direction. And on the right is a curve that shows the cost in real dollars. So we've inflation adjusted these dollars. And what you can see is it's getting more expensive to make each unit gain. And that's clearly not a sustainable system. And the only way out of that trap that I know of is to continue to innovate and bring new technologies to bear on these problems. And then there are a number of bullets here talking about some of the other challenges in agriculture, which you all know. But the one that I really think is important to highlight is increased weather variability with climate change. And so that is, in fact, much of the foundation of the whole G/E project within the Genomes to Fields initiative, right? If we can understand the genetic control of genotype by environment interactions, presumably we can harness that in our breeding programs. So, you know, how do we begin? Well, beginning several years ago, several decades ago now, the federal government has been investing in genomic tools and genomic resources to begin to lay the groundwork to, in fact, breed crops that are more stable across changing climate. And, you know, one of those was sequencing the genome, which was released in 2009. That was a $32 million project. But since then, there's been this absolute explosion of maize sequence data in the public sector. So our challenge really is how do we, or our opportunity maybe, or challenge, is how do we make use of that to begin to, in fact, develop crops for a world in which climate is changing. And one of the approaches, which I'm quite enamored with, is to use the massive amounts of genomic data, because it's very straightforward to generate this now, along with large amounts of phenotypic and environmental data in conjunction with the big data paradigm to develop predictive models so that if we have genotype and we can make predictions about the environment and we understand G/E, we should be able to predict the phenotype. Now, of course, we can't predict the environment with certainty in coming years, but we can make better predictions every year. And if we really understand that equation, it will help us in terms of improving crops. So we would expect that if we're successful at building those predictive models, we'll be able to increase the rate of genetic gain per year to a large extent simply because we will be able to look at the genotype and predict which lines are not worth field testing. So we can focus our field testing resources on lines that are likely to perform well. We can also breed crops to withstand increased weather variability. And we can improve our ability to provide farmers with evidence-based recommendations, you know, which varieties to grow in which fields under which management conditions. And this should lead to both increased yields, but probably much more importantly, increased stability of yield. And then finally, we can also begin to look at public sector predictions of production levels so we can see early signs of food emergencies and deal with them before they become crises. So in order to do that, what do we need? Well, we need an enhanced understanding of how diverse factors, genotype, environment, and the interaction of those two terms influence phenotype. We need data. We need lots of data. The phenotypes are really the rate limiter here, and that was the driver for the G/E project. So it's easy to get genotypes now. There are lots of great tools and sensors out there to get environmental data, but measuring phenotype is still very rate limiting. And the other challenge is management practices. So coming up with standardized terminology so we can capture what was actually done in a particular field in a particular year and include that as metadata in our yield trials. And then we clearly need to overcome that bottleneck of phenotyping. We need new phenotyping technologies, sensors, robots, UAVs, GAVs, and so forth. And then equally important, at least, is new methods for analyzing these massive amounts of data that are coming out of these sensors and robots and so forth. And then finally, we need a community of plant scientists, engineers, and computational scientists with an interest in working on these problems and that know how to talk to each other. So I'm going to touch on each of these points. We'll start on the G/E thing. So this is a stylized curve. We've ranked a number of environments based on the amount of stress, and for that we've used the average yield of all the entries in a trial as a reflection of stress. So the site with the lowest average yield is the high stress environment. The site with the highest average yield, the black line, is by definition the lowest stress environment. And you know, we will find individual hybrids that have different slopes here. So there will be some like this blue one, which has a more flat curve as opposed to the red one. And there's not a right answer here. Depending on where you are and your tolerance for risk, different slopes make sense. If you're in the developing world and your food supply depends on what you produce, you don't want any risk of a serious crop failure at the left end of the curve. And so you're willing to take a more flat curve, give up the possibility of higher yields if you have a great year to ensure that you have enough food, sufficient food, even if the year is very stressful. Where in the developed world, where we have lots of technologies for reducing stress, we can apply fertilizer and irrigation and pesticides, and if all else fails, there are typically various kinds of government programs around the world to keep you from starving if you have a crop failure. You wouldn't want to go with a blue hybrid, you'd like something red. Now, if in fact we produced something like red, somebody would say, well, I would really like it to be stable across environments, and they would like it to look like that green line, right? And not lose any yield. But if we develop that kind of a hybrid, somebody would say, well, how come it doesn't respond to fertilizer and inputs? And they would really like it to go up. But of course, if we did that, they'd want it to be horizontal again, and we'd, all right. Okay, so really the question that we're interested in, or at least many of us are interested in, is the genetic control of the interaction of genotype and environment. And there have been two models in the literature for decades. There's the allelic sensitivity model, which says that the same genes control average trait performance and the plasticity, just you have different alleles. And the structural gene model is the alternative model, which says that there are actually different or distinct sets of genes that control plasticity and average trait values. And there have been a number of modestly sized experiments over the years trying to tackle this question. But Aaron Kusmek in my lab recently downloaded the very extensive data that Ed Buckler and other colleagues that have been working with NAM have produced and made available to the community, which is a fabulous model for how we should be proceeding here, which is to share data, particularly after we first publish it, and use that to conduct GWAS for about 25 traits on the 5,000 NAM rails in approximately 20 environments. And this was recently published, so I'm not going to spend a lot of time on this, but what was an absolutely clear signal was that the genes for average trait values and the genes for plasticity were essentially non-overlapping. They were very distinct sets of genes. So that strongly supports the structural gene model. But this is good news for us, because this means it should be possible to breed for both high average trait values as well as stability, if that's what we want to do, or high plasticity, whatever we want to do. We should be able to breed for these traits at least reasonably independently. So that's good news. The bad news is that if we want to go in and start studying these genes and begin to manipulate them and use them for the benefit of crop improvement, for most of the genes, not just the ones that Aaron found, but if we just look at all the genes in the maize genome, most of them there's no published information at all. So this is a histogram that Eddie Ye in my lab pulled together by mining Medline, and it's a little biased because it only looks at the papers that are available from electronic sources. But out of the 40,000 genes in maize, 35,000 of them or so have no publications at all. We have nothing in the public literature about that. Now you can say, okay, if we go to MaizeGDB, there's some annotation, but as Carolyn Lawrence and her colleagues in her lab have said, most of the annotations that are present are generated by relatively low confidence automated processes. So we don't know what these genes do. And one of the things that I think we should be doing as a community, and that will be one of my contributions to the white paper that Carolyn Lawrence Dill is putting together, is that we need to focus in on genes that are associated with important traits, for example, yield and stability of yield, and begin to understand what those genes do. We just can't rely on associations to human papers that, maybe it is an aldehyde dehydrogenase, but what does it actually do in the plant and why does it matter? Okay, so let's move on to data. And this is what brings all of us here together, is this extraordinary multi-state G/E project. And this is the executive committee for this group is shown here. But this, Natalia De Leon has really done a fabulous job of pulling this community together and making this work. It is truly an extraordinary thing that many of you are involved in. It's generating a data set that is, as far as I can tell, unparalleled in the public sector. And there are a number of groups that have already begun to dig into these data and begin to make interesting and important discoveries. And we're going to see more of these each year as we get more data. As we get more sites, the data become richer and more possibilities exist for beginning to mine these data. But one point I wanted to make here was that most of the data that are collected so far have been done using very traditional phenotyping technologies. And one of our goals, and one of the real reasons for getting together for these couple days, is to really think about how we can bring high-throughput phenotyping technologies into the G/E sites. How can we get various sensors and measurement devices into the G/E sites? And so what do we need, what's out there, and how do we get it? So I'm going to just go through a little bit of some technologies. I'm not going to, I've tried very hard not to replicate what we're going to hear in the next day and a half, though I will introduce some of these. But as we think about what we want from phenotyping systems, and we all probably want somewhat different things, but I'd like to be able to phenotype at multiple time points so that we can, during growth and development. So we can study not only the endpoint, say the final yield, but also we can look at dynamic traits, such as the rate of growth or the way the plant responds to stress. We also need to be able to phenotype entries in multiple environments. If we're going to study environmental effects and G/E, we need technologies which can be deployed over many sites. We can't use some large infrastructure that's only going to be available at one site. We need to have technologies which are fairly nimble and hopefully relatively inexpensive so we can deploy them at, ideally, all of the G/E sites someday. And then for maize, as a geneticist, much, certainly not all, but an awful lot of what's exciting about a maize plant you can only see looking at it from the side. And looking from above, you miss a lot of the exciting stuff. So we need to think about how we can use technologies that will let us look at the plant from the side. So there are lots of technologies out there. I'm going to focus today on just a little bit on field-deployed UAVs, and mostly I'll talk about these stationary field-based sensors, particularly very inexpensive cameras. But I'll start with this Phenobot 1.0, which is a project that was funded by USDA. Lee Tang and Maria Salas are my co-PIs on this project. And it was Phenobot 1.0. It was a very early version. The idea was to get a technology out there. This was a number of years ago, but just convert a garden tractor into a robot by putting an auto steer unit and a GPS unit on it, along with this boom, which you could put pairs of cameras so you could create stereo images from the, this was actually sorghum. But one tripod there and watched birds eating different kinds of bird food, and it turns out that they tend to kick milo right off the feeder, that's sorghum, because it's bred to be not taste good for birds, so they don't eat it in the field. So putting it in a bird feeder has limited success, where if you put proso millet in there, they love it. But anyway, we were watching the birds do this, and I got really intrigued with this. James had been doing some work during his postdoc at the Danforth Center with this technology, looking at corn. But we decided to collaborate a little bit on this, and I made a very significant contribution, which was to build the wooden adapter that connected the Raspberry Pi to the tripod. James had been using duct tape, which actually works pretty well, but it just offended me, so I built this wooden thing. But anyway, we then took this idea that James had shared with us, and we scaled it up and made it field ready. And so I will just show you a little bit of what that looks like. So we had the NAM rails out in the field. Those are solar panels because we didn't have electricity out in the field. And then the poles each have a relatively low-end consumer camera in a, unfortunately, nearly waterproof case. And you can see them there with now the plastic that we would then, the plastic bags that we would put over them every night to keep the dew from going into the nearly waterproof cases. And then here are the Raspberry Pis that are driving these cameras to take pictures every 5 or 10 minutes. We have weather stations there, and then we're able to watch the progression of plant growth and development. So here we can look at plant height. If we scroll through here, we can watch anthesis as it goes through. There are a number of things we can do, but I'm going to just skip ahead. This generates a lot of data. This is not a new discovery, but I think every one of us discovers it ourselves. You generate a lot of data very quickly with these sensors and automated camera systems, so you need a plan to analyze it. And Bhaskar will be telling some of the approaches. I think tomorrow is Bhaskar's talk. He'll talk about how he helped us analyze these data sets. But this has been a challenge with every new sensor type that we've developed. We end up with an awful lot of data. And I don't think that's a bad thing. I think I'd rather have too much data and struggle to analyze it than not have enough data. I think sometimes having a hard drive full of data is a stimulus to actually coming up with better ways to analyze it. But we've also deployed these cameras to do the time-lapse photography out in western Kansas and western Nebraska so that we could actually get droughts. We don't usually get droughts in Iowa, and I always felt bad hoping for a drought here because it would reduce the tax base, and my salary depends on that. So I figured we'd go to the states to the west where I could hope for a drought. Now that James is in Nebraska and one of my other students is in Kansas, I feel a little bad hoping for drought out there, but less bad than for Iowa. But the idea is doing managed stress or even non-managed stress, looking for drought tolerance is a very expensive process. There's a limited amount of field space, and it takes a lot of management to do these. So we're asking, with the camera, can we see some feature of the plant as it responds to stress that would be a predictor that would allow us to just do a first cut to remove some lines and not test them at all? Or maybe there'd be some signal that would say, these are lines that actually look quite interesting. For example, since we've got both irrigated plots and non-irrigated plots along with the camera plots, we can ask, are there lines that do well in non-irrigated, so they're drought stressed? Can we see something on the cameras for those lines under drought stress that gives us a clue that they're actually responding well or tolerating that drought? Or maybe it's the difference in yield between the irrigated and the non-irrigated. Maybe we can find a signal from the way the plant grows that will give us a clue as to which lines actually tolerate the drought by not losing a lot of yield when they go under drought stress. So without going into a lot of detail, we've been doing this for a couple of years now. We've collected a lot of data. Every time we get a curve that looks like that left, it's a sigmoidal curve if we're just plotting the growth rate over time. That plot is a series, I think 5 or 6 plants of the same genotype that are all growing in one row. And we see variation from plant to plant as we would expect. But we get a very consistent pattern as we start looking from genotype to genotype. So if we look over on the right, we have 2 genotypes in 2 locations. And what we see is that among genotypes within a location, we see a lot of differences in growth rates. We also see differences in growth rates between locations within a given genotype. And this is clear evidence for genotype by environment interaction for growth rate. And so now what we want to do is start correlating what we're seeing in terms of growth rates at multiple locations, lots of genotypes, correlate that with stability across the G/E trials, because we've got some of the same lines in both of these experiments. And tomorrow, I believe you're going to hear some ideas from Liang Dong, who's going to talk about why we see difference in growth rates among locations and among genotypes, particularly under drought stress. So I want to now talk about sort of a philosophical approach to phenotyping. And I was having a discussion with a student at the poster session, and I said something. We were talking about whether or not there was actually a design for G/E, whether there were hypotheses. And there certainly are some hypotheses, but also there's just a real effort to generate a lot of data with the strong belief that having those large data sets is going to allow us to extract understanding of the plant from those large data sets, even if we haven't necessarily designed a hypothesis-driven experiment. And the point I want to make today, and it may not be as hard to sell with this group as it is with some groups, but that there is a role, certainly hypothesis-driven research has been very important and valuable to the community, but there is also a role for data-driven research where we generate lots of data and then we look for patterns in those data, even without going in with a hypothesis. And so that's the point I want to make in the next couple slides. So we can do hypothesis-driven research, we can do data-driven research. That can be in controlled environments, it can be in field or target environments. These are complementary. They're not in competition. So this is back in '85, we began doing some work with UAVs. And we had that large planting of the NAM lines. We also had a diversity panel we call the SAM panel. And we flew the UAV over and we just collected data. And we, again, didn't have a hypothesis for that. That was really just testing out the UAVs, trying to get comfortable with the technology. And we learned how to collect the data and clean it up so that we could use it. And we started noticing patterns, though, in the data. And I want to talk about one of the patterns that we found. So this is the work of Ting Zhang, who was one of my graduate students at China Agriculture University, and she came over for the summer to collect data for her master's thesis. But so while she was here, she collected UAV data and then started looking at it. And if we look at the axes of the plants in the left, they are all at different angles. And this is exactly what we would expect based on the way we planted. This was, I'm not actually don't remember whether these were jab planted or machine planted, but the kernels go in and not random perhaps, but pseudo-random or pseudo-random orientation into the soil. And we know from the middle panel that the orientation that the kernel goes into the soil absolutely defines the first leaf, the orientation of the first leaf. And the second leaf is opposite that, and the plant does this. So we would expect, since the kernels go into the soil in a random orientation, we would expect exactly what we saw on the left. But we saw in a number of rows this phenotype we called plants parallel, which is on the right. So this was not an experiment that we set up. We didn't set up looking for this. We just flew the UAV over and we started seeing some interesting patterns. And there were a number of genotypes that had that plants parallel phenotype. Well, so is this just noise? So I asked Ting to run a GWAS, and she did, and she found a number of hits, which suggested that this really wasn't noise. And we've since gone on and established that it is not noise. But how do we go from a randomly oriented kernel going into the soil to plants that are perfectly oriented with each other in a parallel way? Well, we reasoned that the plant must be reorienting itself during development, and so we tested that. This is the work of Yan Zhou, who's been working on this project since Ting went back to China. And if we just look at this plant up here, we can see that, in fact, these plants do have, some plants, not all plants, some genotypes have the ability to rotate during development. You can see that leaf swinging around. So we started with this data-driven approach. We developed a hypothesis that was in the field, hypothesis which we tested in a controlled environment, the greenhouse. Then we moved back out to the field where we now oriented our cameras from above, where we could better watch the plants out in the field and watch whether they were turning. And we're studying the behavior of individual plants over time, asking about the genetic control of that, as well as how these behaviors may be influenced by neighboring plants in the environment. And one hypothesis we considered was that the orientation of the sun was somehow, there were some plants, some genotypes were aware of the position of the sun and using that to entrain themselves and become parallel. Well, we were able to rule that out simply by observational studies. So if we look in panel E there, that's the border of our nursery, and it's planted with a pioneer hybrid, as it would be, Matt. And what you can see is those plants are parallel with each other. And as you go around the curve, around the corner, they stay parallel. So they're running north-south, and then they're running east-west, and they stay parallel in both directions. So it's not simply that they're recognizing where the sun is. I believe they're probably recognizing their neighbors through some process, and I don't know what that is yet, but that's an area of very active investigation in the lab. So this is, as I said, a data-driven approach which led to hypotheses, testable hypotheses. But it also begins to fit into what we saw in one of the very first slides this evening, which is that green panel down there in the lower left, which is that yields have been going up. And if we look in the bottom panel in the middle, some of Don Duvik's work, we can see that what happened during this period of increasing yields has been increased plant density, that the average yield per plant has not gone up very much. What's really happened is we've been able to pack more plants into the field. And maybe the reason we've been able to pack more plants into the field is because they are now lined up like little soldiers in the field. So it's just like a Tokyo subway or something, right, where you pack as many people in as you can, or a crystal lattice. So that's something we're quite interested in looking at. Now, the only thing that doesn't fit with that hypothesis is B73 doesn't turn. So we need to continue to follow up on this. Okay, so I want to finish up with a topic that I think is extremely important. Having lived through the bioinformatics era, so the beginning of genomics and the flood of data that we got, and the recognition that most biologists just simply weren't prepared at that time to handle the large amounts of data that were coming out, and we had to reach outside of our community to bring computational experts in to the field to help us with that. I don't want us to make that same mistake again. I want us to plan for this, recognizing that we are being buried in data and that phenomic data are much more complicated to work with than genomic data because they're so much more variable, and of course there's a lot more of it. So how do we do this? We need to obviously have the plant scientists involved. We need engineers who are excited and interested in building sensors and deploying sensors on UAVs and getting them out to measure both the phenotypes and the environment. And we need computational scientists with interest in designing and analyzing these very large experiments, and often developing new algorithms to deal with them. So as we thought about how to do this, I was inspired by Jim Cornett, who was a mathematician here at Iowa State, who started coming around to the molecular biologists in the early 1990s. And he volunteered to introduce us to various computational scientists on campus, the mathematicians, the statisticians, the computer scientists. And in the early 90s, you know, we were very polite, I think. I tried to be polite, but Jim, we really don't need any computation. I mean, the band is on the gel or the band isn't there. We don't need anything. But by the mid to late 90s, it became clear we needed help. And so when he tried again, and I thank him for doing that, we were quite... We welcomed this opportunity with open arms. So what Jim did was he brought together every week biologists and computational scientists. And we alternated weeks where the biologists would talk about the data sets we had and our challenges with trying to analyze them. And in the alternate week, computational scientists would talk about various algorithms that they were aware of or that they had developed and that could be perhaps brought to bear on some of these problems. And for me, it was a career-changing semester. It was one of the most fun things I'd ever done. And it really, it changed campus. It created all of these fabulous collaborations, and it just changed the whole culture, and it changed how we trained graduate students. We got one of the I think it was the first IGERT in bioinformatics anywhere in the country from NSF, the training grants. It was a really significant thing. So we've deployed the Cornett model for building a predictive phenomics team at Iowa State. We began with a workshop where we invited the plant scientists, the engineers, and the computational scientists. We met for 2 hours every Friday during the fall of the 2014 semester. And you know, there's this old line that if you bring pizza, you can get graduate students to do anything. It turns out pizza doesn't work for faculty, but gourmet sandwiches do. So we got some very nice sandwiches, and people came. We had only internal speakers, and it was only faculty, so that it was like a lab meeting type of environment for faculty. There was nobody else there, so people could ask the so-called dumb questions, but they didn't seem dumb. It was really an extraordinary semester. I had a real blast, and we did we had about 30 faculty who attended essentially every week for the whole semester. And you know, you all know it's really hard to get faculty to go to anything more than a couple times, but they just kept coming. The evaluations were extremely positive, and it was. It's something that can be replicated at any campus. I guess that's the point I'm trying to make here, which is you can all go back and do this, is engage the engineers on your campus and the computational scientists and try to bring them into the fold, because we need more ideas, we need more people from outside of the plant science community itself. We need a much broader sampling of different disciplines. Now, I was fortunate because around this time I was appointed director of the Plant Sciences Institute, and so I had some funds that were available, and I was able to redirect more funds. So I had done my my postdoc. I was a I was a NIH fellow at the Max Planck Institute in Germany, which uses a system where they pick rising stars of faculty and they make them directors of the Max Planck. And they give them enormous resources, and they just basically let them do whatever they want. They just pick people. So pick people, invest in people rather than projects. And it's the same model that's used by the Howard Hughes Medical Institute, is pick people. If you can do the picking, that's the key. You have to pick the right people. But if you pick people that are just going off like this, give them resources and let them have to it. So we went through a process of picking PSI scholars. We were able to fund 16 of them from a diversity of disciplines to build this community. And then gave them relatively modest but transformative amounts of funding that was stable over several years, and they could spend it on anything they wanted, anything. There was no rebudgeting. If they wanted to buy a piece of equipment, hire a grad student, a postdoc, take a trip, go on a sabbatical, bring a visiting scientist, anything you want to do, you can do it, as long as it's legal, and that's what my assistant keeps track of. And it has been, I would say, very successful. The first cohort started in February of 2015. We, as I said, brought 16 in. As of half a year ago, they'd published 136 papers. So that's 2 years of data, just 2 years of data. They'd published 136 papers. Most of these people were not working in phenomics when we began. So this is not simply counting the productivity of good scientists. This is stuff they were doing that they hadn't been doing before. Lots of speaking engagements. My central administration worries a lot about this return on investment. So for every dollar we've invested, we've brought in $4 of extramural funding. That's a good ratio. And then there's the second cohort, which will be announced this week. We're going to bring 9 more PSI scholars in. So the first piece, the workshops, every one of you could do this on your campus. This one takes some money, but if you talk to your development officer, maybe you can convince them to find some rich person who would like to fund something like this on your campus. I think it would be amazing if we could do this at some other campuses. And then just a couple of graphics to show how this works. So we have the plant scientists, the engineers, and the computational scientists color-coded. And these lines are true collaborations, where there's a grant proposal, there's a paper, there's an actual project. And essentially none of those lines existed before we began. And so we've built this whole network. And it doesn't just affect the people who are funded. The white dots here are PSI scholars, and then they're reaching out to other faculty on campus, building additional collaborations. So it's really changing the whole culture on campus. And I haven't shown here off-campus collaborations. Those are obviously often quite important. So along the lines of changing culture also is that we can bring computational folks and engineers into the fold of plant science, but we also need to think about the next generation, and can we train them so that they are ready for this new world? I don't think we can train very many people have the ability to be a true engineer and a true computational scientist and a plant biologist, but we can train people at that intersection so that the plant scientists are able to communicate with computational scientists and engineers. They'll understand what's possible and how the reward structure works in different disciplines, which is extremely different, and it's important to know that. So we were able to get an NSF training grant at this intersection. This is led by Julie Dickerson, who's the PI, and we're training PhD students right at this intersection. So if you have undergraduates in your lab who are interested in this area and have computational abilities, they're quantitatively oriented, please encourage them to apply. We're really looking hard for US citizens and US nationals, permanent residents, for this program. So with that, I will finish and answer any questions you might have. Thank you. Questions?
Pat, was there any correlation between the genotype's ability to pack tightly by orienting its leaves and yield?
We don't know yet. We don't know yet.
Why? You didn't measure yield?
We didn't measure yield on this. We're looking at we're not looking at, the diversity panel that we used is the so-called SAM panel, and it has a lot of stuff. Some of it's very commercially relevant, but there's stuff that isn't. But we're headed there. Others? I'm not going to let you leave, until we get at least a couple more questions. Yes?
So I like the approach that you explain and put together here at Iowa State University. My question is, what parts of this approach, bringing people together, would you apply for the EU, for the G2F project?
Well, that's partly what we're doing here these 2 days. So you're going to hear tomorrow and the next day from a number of engineers and computational scientists. And so I hope we all use the limited amount of interaction time that we have to in fact start building those relationships. But what I really hope is that each one of the Genomes to Field sites starts thinking about doing something like this on your own campuses, because there's no reason you couldn't do it. The first part, the workshop, the Cornet model. It really could be done. It requires probably getting buy-in from administration to sort of stamp of approval. But what we found is people really enjoyed it. There's just so few times when faculty can just sit back and think about things that we don't normally think about that are scientifically oriented, right? I mean, most of the time we spend, I mean, I don't know about you, but I certainly spend an awful lot of time doing stuff that I never would have thought I would be spending time on a long time ago, right? And so it was just a pleasure to spend 2 hours a week listening to smart people talk passionately about what they knew and cared about, and then start thinking about, oh, wow, God, I wonder if I could use that in my research. It was really fun. So I would love for all the Genomes to Fields collaborators to try it. Yes, Nathan?
So Pat, it seems, as you go through the different technologies and platforms you've used just over the last 5 years, I get the impression year to year, what you're measuring, what sensors, what platforms are changing. How much of your data can you go back through time? You say the traits I measured in 2017, I can compare them to 2015. Or are you finding platforms are changing so quickly that the measurements are hard to compare across years at this point?
Yeah, that's a good question. It's a topic you and I usually discuss, sort of stability versus change. So what we've been doing, like for the cameras, the time-lapse photography that we've been doing that fairly consistently for several years, since 3 years now, out in the field. And what we're doing is we're layering different sensors on top of that. So we still have the base, the consistency of the time-lapse photography, but we're adding sensors, some of which you'll hear about tomorrow, to measure. You'll hear about them tomorrow. Cool stuff. But we are not in a state where we can just say, this is going to be the VCR, and we're going to stick with the VCR from now on, because somebody's going to invent a DVD. But yeah, one way to deal with that is to have at least a few years of overlap, so that you can compare. Yes, Seth.
So your idea of cross-collaborative sharing is really nice, and I think there's a lot of groups that have been working on that. But one of the challenges that I've seen is that if you're working with cutting-edge statisticians, computer scientists, mathematicians, for them to succeed in their discipline, they have to develop principles in their own basic research and their own basic field. It's a lot harder sell to bring them to work on your applied problems, right?
Yes.
So do you have some examples of success for that, and how have you developed those things where you actually, brought very basic researchers from those areas into applied problems?
Yeah. So Dan Nettleton, who's sitting 2 seats over from you on the table, is a world-renowned statistician, and he meets with my team, or we meet with him every week, unless my students cancel it. We have essentially a joint lab meeting, and mostly it's my students asking questions about experimental design or how to analyze some unusual data sets, which would fit into your sort of applied piece. And the reason Dan tolerates us, I think, is because once in a while, a couple times a semester, we come up with some problem that isn't, oh yeah, here's how you do it, and it gets him thinking about some new problem that he can give one of his students and get them started on the cutting edge thing. So this is what I meant also about the different reward structures in the different disciplines. It's exactly that. I mean, you can't expect really cutting edge, talented faculty to just come be, your tame whatever. So there has to be this relationship where, we're willing sometimes to generate more data or different sorts of data that will help, in one case, Dan, test some statistical models. Yep. Yes, Steve?
So Pat, as you get more and more phenotypic data, do you reduce all your data to simple data types so that you can make correlations? Or are we going to get to the stage at some point, where we have ways of being able to correlate different phenotypic information without, let's say, reduction to
numbers? Yeah. My guess is number 2, but I think we'll hear about that from folks tomorrow. I will say that I think it's important to keep raw data. We used to talk about how expensive it was, and I've told this story to a lot of people, so if you've heard this before, you'll just have to listen to it again, which was that in the era of Sanger sequencing, there was one major seed company that had gotten in early and done a lot of EST sequencing. And you remember we got these trace files, right? A lot of you probably don't remember this. Some of you may have seen these in historical texts. They're 4-color trace files. And there were ways of taking those trace files and converting them to A's, G's, C's, and T's. And at that point, it saved a huge amount. You're getting rid of an image, and you're just saving A's, G's, C's, and T's. And so this company did that. They threw away all the trace files and just kept the A, G, C, and T. At which point, somebody came up with a better algorithm for converting trace files to nucleotide sequences, and it wasn't possible to go back because the raw data were gone. And that's what I don't want to happen with phenomics data. Now, some people will argue, well, you know, you could just regenerate the phenomics data, but we're never going to have a year 2017 again. So that was a unique sampling of environment, and we're never going to get it again. So I'm keeping all of the image files. Everything is being kept. I used to worry about storage, but James just told me that you can store it on Amazon Glacier for, I don't know, 10 cents a gig or something per year. I mean, it has gotten very inexpensive if you don't need quick, immediate access to data. There are ways to store large data sets. Yeah, Madan.
You mentioned in the beginning that there is a huge problem about functional characterization of the genes because probably 80% of the genes are with unknown function. Do you have any thought on that, how we are going to solve that puzzle?
The way I would like to solve that, thanks for that question. The way I want to solve that is we're not going to do it with all genes, but find genes that are associated with important traits, so genes that we actually care about, and then go in and mutate them so that we can study their effect. And for example, with Aaron's experiment, right, we found genes that are associated with plasticity. It would be fabulous to make mutants in those genes and then deploy those mutants at the genomes to field sites, the G/E sites, right? So we can actually look at, if we make a mutation in this gene, whether it's a transposon or a genome edit, we have variants and deploy them at multiple sites and then carefully measure them with some of these new sensors that we're developing and ask what the effects are. But it would take a community to do this. At least I think it takes a community to do that. But it'd be a fabulous experiment for us. Other thoughts? Seth again.
So NCBI has a great resource for storing some of the genomic data once you've extracted it from trace files.
Yeah.
What's the thought right now on where we should be storing the raw images from?
Yeah, you and I have spoken on exactly that topic, and I've asked so many people about this, and I don't think there is a good solution yet, and so maybe that's something we need to put into the white paper, and maybe it is partnering with places like Amazon and just having a budget for storage. You know, it's not reasonable that, I mean, for libraries, right? I can go back and get the experiment station report from 1910. It's in the library, but somebody's been paying to store that. There's been a building, there's been electricity costs, there have been curators, but we decided in 1910 we were going to start keeping that stuff, and it's there when I need it, and maybe we just need to decide to do that.
But I will just point out that for genomic data, there is a federal data
Yeah.
resource, right? And
Right.
there is support for that. And
There is, and it's been fabulous. And USDA has really stepped up and been the major player for the long-term storage and curation of those data. The phenomic data, as we all know, is much bigger and much more complicated. But maybe that is a role that USDA would like to take. We need to find some partner to do it because it would be an absolute shame. And we know this happens. If we leave the data in individual labs, students, postdocs, staff leave, eventually faculty leave, the data will be gone. And it doesn't actually take very many years for things to disappear. So, somebody, Jimmy.
I like your example about this data-driven, then generate new hypothesis. Can you share more insights about, you know, when we do this data mining, essentially we try to find a pattern.
Yeah.
In that particular case, was there careful observation to find the pattern first, or there's a systematic pattern finding algorithm to apply or anything like that?
I wish I had a story about some great algorithm that we had. It was a number of people looking at videos of UAVs flying over fields. And the people that were looking were very early stage graduate students and an engineer. And so they came up with a lot of patterns, which I, as a biologist, decided didn't warrant further investigation. But they did come up with this one. And as soon as I saw that, I said, that's interesting. And so we followed up on it. I suspect eventually there will be more automated processes for this. Yeah. Anything else? Yes, sir.
You mentioned that there will never be a 2017, for example, and your paper with Aaron Kuznick goes into the differences and different stresses in the environments. What efforts is G2F using to be able to quantify those environmental differences that you're experiencing at all of these different sites that you're doing over multiple years?
Yeah, that's a good question. And so there are 20, 25 sites, and each one of those has the same weather station. So we're measuring at least some environmental parameters with the same technology at every site, which is a great step. This was the, I think probably David Ertl and the Iowa Corn Growers paid for all of those. I'm not sure, but they paid for a lot of them. But the decision to use the same instrument at all the sites was a really good one. The decision to, for example, we just got an email, I think all of us got an email recently to check our weather stations and make sure they're working properly and calibrate them before they go out into the field. That's a good idea. We still sometimes get some pretty weird data. Somebody's changed the cable that's supposed to go in the red socket with the blue socket, and you start getting weather, temperature in the rainfall column and vice versa. Those can be easily fixed, but we need to be very careful and measure. There are many other parameters we could collect. And I think another thing that can come out of the white paper in the next 2 days is really start talking about what we need to make the G/E platform even more valuable to everybody. Expand it and also measure more things. Anything else? All right, well, thank you very much, and we start early tomorrow. I don't know, what time is it, 7:30? Breakfast? Somebody know? Yep. Yes? Yes. You're sure? Okay. 7:30 here. Is that right, Marianne? 7:30 here, breakfast, and then we start with talks at 8 o'clock. Okay. Have a nice rest.